We're continuing with cell biology and life science in a nutshell. Um, we were learning about um, carbohydrates and uh, fat cells as well, and how um, these are converted into one another um, via a special cycle. So when carbohydrates are difficult to come by for organisms, they break down fatty acids in order to derive subunits for carbohydrates. So we learned that the glyoxylate cycle is used in order to produce glucose, which are subunits for carbohydrates from fatty acids. This process is present in organisms, algae, cyanobacteria, plants, and fungi primarily. So this does not occur in animals. So gluconeogenesis, which occurs via the glyoxylate cycle, is a metabolic pathway that results in the generation of glucose from certain non-carbohydrates, and this occurs in non-animals, as shown here. So in the glyoxylate cycle, simple carbon compounds are used to synthesize macromolecules, including glucose. In this way, fatty acids from lipids are commonly used as an energy source in order to um, generate the molecules that are needed. And energy is obtained from fat using the glyoxylate pathway. Acetate from fat is utilized for the biosynthesis of carbohydrates in the glyoxylate cycle. The initial stage reactions of the glyoxylate cycle are similar to the TCA cycle. So we'll touch on this in a bit. So cell wall containing organisms such as plants, fungi, and bacteria require large amounts of carbohydrates during growth for the biosynthesis of complex structural polysaccharides such as cellulose, glucans, and chitin. In these organisms, in the absence of available carbohydrates, for example, in microbial environments or during seed germination in plants, the glyoxylate cycle permits the synthesis of glucose from lipids via acetate generated in fatty acid beta oxidation in the glyoxylate cycle. The glyoxylate cycle bypasses the steps in the citric acid cycle where carbon is lost in the form of CO2, carbon dioxide. The two initial steps of the glyoxylate cycle are identical to those in the TCA or citric acid cycle. Where you have acetate to citrate to isocitrate. And then in the next step, catalyzed by the first glyoxylate cycle enzyme, isocyclate lysase, isocitrate undergoes cleavage into succinate and glyoxylate. The latter gives the cycle its name. Glyoxylate condenses with acetyl-CoA, the step catalyzed by malate synthase, yielding malate. Both malate and oxaloacetate can be converted into phosphoenol pyruvate, which is the product of phosphoenol pyruvate carboxykinase, the first enzyme in gluconeogenesis. So, 5 of the 8 enzymes associated with the TCA cycle are used in the glyoxylate cycle. These are citrate synthase, aconitase, succinate dehydrogenase, fumarate, and malate dehydrogenase. The two cycles differ in that in the glyoxylate cycle, isocitrate is converted into glyoxylate and succinate by isocitrate lyase instead of into alpha-ketoglutarate. This bypasses the decarboxylation step that takes place in the TCA cycle, allowing the simple carbon compounds to be utilized in synthesis of macromolecules like glucose. Therefore, the glyoxylate cycle is a variation of the tricarboxylic acid cycle, TCA, and it is an anabolic pathway occurring in plants, bacteria, protists, and fungi. It allows cells to utilize simple carbon compounds as a carbon source when complex complex sources such as glucose are unavailable. The cycle is generally absent in animals. The exception is in nematodes at early stages of embryogenesis. In recent years, the detection of malate synthase and isocitrate lyase, key enzymes involved in the glyoxylate cycle, were detected in some tissues of animals, raising questions regarding the evolutionary relationship of enzymes in bacteria and animals, and suggesting that animals encode alternative enzymes of the cycle, which may differ slightly in function. The citric acid cycle takes place in the inner mitochondrial membrane of animal cells. On the other hand, glyoxylate cycle occurs in plants, usually in germinating cells, etc., and in microorganisms. This is called gluconeogenesis. So in mitochondria, you have fatty acid beta oxidation. So the TCA, or the tricarboxylic acid cycle, or the Krebs cycle, is a series of chemical reactions used by all aerobic organisms to release stored energy through oxidation of acetyl-CoA derived from carbohydrates, fats, and proteins into carbon dioxide and chemical energy. It is also called Krebs or citric acid cycle. It is the second stage of cellular respiration. It is a 3-stage process by which living cells break down organic fuel molecules in the presence of oxygen to harvest energy they need to grow and divide. It is a series of enzyme-catalyzed chemical reactions forming a key part of aerobic respiration in cells. This occurs in the mitochondria. And here we have a carbohydrate such as cellulose.